Um, so welcome, Natalie. I
Thank you.
think this is a good time to start. So please welcome Dr. Natalie Stroeymeyt here from the University of Bristol, and she's here to talk about how ants might reduce epidemic risk by altering their social networks, something that's really exciting. And I can't wait to hear what she has to say about that. She has done her PhD in neuroscience, behavior, and cognition from the University of Bristol and the University of Toulouse on information gathering prior to immigration in house hunting ants with Professor Nigel Franks and Martin Joshua. And she went on to continue with her passion for ants in her postdoc as well with Professor Laurent Keller at the University of Lausanne. She has done some really amazing work on disease spread in ants and how they spatially organize to avoid such spread. I have heavily cited her during my PhD thesis for sure. And they've developed this amazing technique and perfected it over time where they've tracked successfully entire ant colonies. Yeah, and so it has led to some amazing results and findings that wouldn't have been possible without it. So yeah, I'm looking forward to hear more from her. Over to you, Natalie.
Thank you very much, Nikita. I hope you can all hear me. Okay, if so, I'll get started by sharing my screen. Okay, so hopefully now you can see my PowerPoint. Okay, so thank you very much for the invitation and for giving me the opportunity to talk to you today. And as Nikita announced, I'm going to talk about social network plasticity and disease transmission in the ant Lasius niger. So as you can maybe see from this picture, ant colonies are one of the most well-known examples of social life. And it's usually thought that their sociality is one of the reasons for their massive ecological success, because being social allows them to cooperate and to work together in order for the colony to do well. But being social does not only come with benefits, it also comes with a series of costs. And one of these costs is the increased risk of transmission of disease. And this is particularly the case in social insects because individuals have a very high genetic relatedness so that they may be susceptible to the same disease. They have extremely close and frequent interactions, they exchange food, so they have a high likelihood of transmitting infectious pathogens between individuals. And then they live in nests which are tightly controlled for temperature, humidity, which is basically a pathogen's heaven. So in theory, ant colonies are quite favorable to the transmission of infectious disease. And yet, if you go into the field and try to look at naturally occurring ant colonies, you very rarely find colonies that are really ill. And one of the reasons for that is that they have evolved a series of mechanisms to try and counteract this increased risk of disease transmission. And altogether, these cooperative mechanisms provide a form of social immunity, which is a concept that was coined by Sylvia Kramer in 2007. So social immunity can take a variety of forms, and some of these are linked with nest hygiene, so trying to prevent the entrance of pathogens in the colony. So at the top left, you can see a garbage pile by leaf-cutting ants. So ants tend to separate their waste so that they are less likely to come in contact with opportunistic pathogen growing on these piles. The bottom left, you can see a cemetery. So in a similar way, ants tend to gather their dead bodies outside of the nest in a very concentrated pile, again, to avoid the risk of being exposed to pathogens that grow on dead body. And then they may also mix up some antibiotic compounds with their nest material, which you can see on the top right, with an ant sort of transporting resin, which has antimicrobial properties. And so this is mixed up with the material constituting the nest and decreases the risk of a disease starting. Then ants also have a series of mechanisms that allow the healthy individuals to take care of pathogen-exposed individuals, such as grooming of pathogen-exposed individuals, which allows them to basically remove infectious particles that are on the cuticle of the exposed individuals. And that also allows them to decrease the risk of these individuals becoming ill and developing disease. Alternatively, they may switch to a kill or exclude strategy where they will deny entrance from infectious individuals. And then one of the less well-known but recently sort of highlighted mechanism is that they can also use their social organization and their network of interaction in order to decrease the risk of epidemics. And this is a hypothesis that was sort of suggested some time ago, and which was called organizational immunity. And the way this worked is basically based on the fact that insect societies don't have a random social organization, but they have a very sort of defined organization in which you have an age-related division of labor among workers. So typically, after hatching, young workers will do tasks such as taking care of the queen or taking care of the brood. And that's happening at the core of the nest. So young individuals tend to be nurses. And as they grow older, they will move towards the periphery of the nest and take on tasks such as nest maintenance or nest building. And finally, at the end of their life, they will leave the nest and perform the more risky task of foraging and nest defense. And because these different tasks take place at different places in the nest, the result of this division of labor is that interactions don't occur at random in the nest, but you tend to have groups of individuals that interact more often with one another than with others. And so you get these non-random interaction of networks, which may have some consequences for epidemics, basically. So this idea of organizational immunity is that the structure of the social networks provides some degree of protection against the transmission of infectious disease within the nest. And the first part of this protection is one that is prophylactic, which means that is always present in this sort of basal organization of the nest. And this cartoon on the left sort of illustrates the idea. So in the center, you can see the queen in red, and then the young workers in green with the brood in purple. Towards the periphery, you have the middle-aged workers in blue, and then the old workers in yellow. And each node represents an ant, and each edge or link represents a contact between an ant. And you can see that we've represented the fact that there are more interaction among individuals of the same age group than between groups. And the idea is that this should provide some form of protection by creating some bottleneck effects between groups, so that if an infection arises, say, in an old individual, it will have a lower probability of transmitting to individuals from other age groups simply because there are few interactions between these age groups. So the idea is that this overall organization into separate groups should decrease the dynamics of spreading of epidemics through the colony. And then another hypothesis that had been formulated was that potentially there could also be a form of induced protection against disease, potentially via network plasticity, so changes in social interaction that could reinforce this protection. And to give you some examples from our current experience, you can sort of imagine that in humans, the ways that we can change our social interactions when a disease is present can take many forms, such as the isolation of the infectious individuals, which can be passive, simply by being sick. Usually we tend to stay home, not move very much, and we don't meet as many people. It can be active, so voluntary isolating ourselves not to infect others, or it can even be enforced with this quarantine that are imposed on sick individuals. Then conversely, healthy individuals can notice that other individuals are sick because symptoms are often visual, and then actively avoid those, which would also sort of change social interaction. And finally, what we've been doing a lot over the last year is a form of proactive social distancing, where everybody sort of spends less time with other people in order to decrease the risk of transmission in case someone actually carries the disease. And all these sort of form of change in social interaction have the potential of reducing disease transmitting contacts. And so the idea was that perhaps the ants could do something similar. And this is illustrated here with these dashed lines showing that the ants could potentially change some of their contacts to further reduce the risk of epidemics. And this could be either via isolating the infectious individual or by decreasing connection among other groups inside the colony. Now, on the other hand, the ants are very well known for their ability to give care. I mentioned that earlier. When an individual is infectious, others will try and save her by removing infectious particles, thereby potentially increasing their risk of becoming infected. So we have this sort of tension, this trade-off, and it's not very clear what happens in an ant nest that becomes exposed to a disease. So during my postdoc at the University of Lausanne, I set out to test these big predictions of prophylactic and induced organizational immunity in order to see whether they indeed were real processes in ant colonies. And to do this, I used the model system of the black garden ant, Lasius niger, and the generalis entomopathogenic fungus, the generalis insect pathogen, Metarhizium brunneum. So Metarhizium brunneum is a fungus that produces conidiospores, which has a sort of transmission form. And when an insect comes in contact with the conidium, that conidium sort of becomes first loosely attached to the cuticle. Then within about 24 hours, the conidium will sort of germinate to form a second cell called apressorium, which will become very strongly ingrained in the cuticle so that it can't fall anymore. And then a few hours after that, this apressorium will form a penetration peg, which will enter the cuticle. And once the fungus has entered the body of the insect, it will start multiplying, forming some blastopores, and eventually kill the insect. This is an obligate killer. So in order to transfer, it has to kill the host. When the host has died, the fungus will sporulate, so form new conidiospores on the outside of the body, very large amount of it, so that when another insect comes close, it will be able to transfer the conidia to this new insect and sort of start over. So the question we wanted to know was, if an ant picks up some of these conidiospores outside from a dead body that's sporulating, what are the dynamics and the risk of exposure of other individuals inside the colony, and how does this relate to their social organization? So to do this, we also needed a way to quantify the social organization of the colony. And we did this by using an automated tracking system, which was based on individual marking of individuals with QR-like barcodes, the fiducial markers. Each of them contains a unique combination of black and white squares, which encode a unique identity. So each ant is tagged with one of these labels on its back, which provides information of its identity and its location. And then the colony is placed in our automated tracking system, which allows the detection and in real time of the position and orientation of all ants in the colony. So we typically have a temporal resolution of 2 images per second. And this was the sort of typical nest that we would use, seen from the top. So on the right-hand side, you can see the nest area, which was actually dark for the ant because it was covered with an infrared passing filter, which means only infrared light goes through. Now, tracking was done in infrared light, so we could see the ants through this filter, but for the ant, everything was dark inside the nest. And you can see the queen, which is a bigger ant with a bigger tag towards the center of the nest area. The brood is shown in white. And then there was a sort of wall connected by a small entrance to a foraging arena on the left, which the ants could enter freely. And they had access to sugar water and water ad libitum in this foraging arena. The advantage of this technique is that by just tracking the ants for quite some time, we can distinguish the nurses from the foragers, simply because the foragers will be the ants that occasionally leave the nest to go and collect food, then come back, whereas the nurses were the ones that always remain inside the nest. This is a video of the tracking system showing you the detection. So each ant that is detected is highlighted by a white dot, and its identity is shown in green next to that ant. And then I sort of highlighted the contact by yellow lines between the ants. So we can do that over an extensive period of time. And then we can use our records to infer the physical contacts between pairs of individuals. And the way we do that is that we use our information on the location and orientation of each ant to represent that ant with a simple shape, so a trapezoid. And the idea is that if there's an overlap between the two trapezoids of two ants, then that indicates that there's a physical contact between them. So we can, in this way, retrace all the contacts happening during our entire experiment, and then use this contact sequence to recreate the network of physical contact for all the colonies. And so you're going to see this sort of graph quite a lot of times. So like earlier, all the nodes represent individual ants. Any link between nodes represents a physical contact between two ants. And the darkness and thickness of these edges is proportional to the frequency of contact between these individuals. Also have a color code, which I will keep for the rest of the talk. So the queen is shown in purple, dark, almost black in the center. The nurses are shown in green, and then the foragers are shown in yellow. And I'll explain later what these black dots are on the foragers. So we can therefore retrace the network of interaction of the colony. And the first thing that we wanted to do was to go back to this idea of organizational immunity and the prophylactic organizational immunity hypothesis, which predicted that the structure of the ant colony with this age-based division of labor should basically protect the queen and the young workers from pathogens coming from the outside, simply because they are the sort of final, most isolated community inside the nest. And so the first thing we wanted to check was whether indeed the queen and nurses are less likely to fall ill from the pathogen if the pathogen first enters from a forager. So we set up this long-term survival experiment where we had 11 colonies, which we tracked for 4 days just to give them time to acclimatize and get an idea of the structure of that network in healthy condition. And then we sampled 10% of the workers from the colony, which we chose among the foragers. So we just picked them from the foraging arena. And we exposed them to live spores of the pathogen, Metarhizium brunneum, which were suspended in Triton X. We then returned these foragers, infected foragers, to the foraging arena, and then continued tracking for 9 days. And during these 9 days, we monitored the survival of these ants so that we could determine which ants were likely to die from this manipulation. And this is the results that we have. So you can see on the x-axis the days after treatment, with 0 being the moment where we expose the ants to exposure. So the minus value, the negative values corresponds to the day before treatment. And then you can see the survival of the treated workers in full line and of the untreated workers in a dashed line. So first, what we found, which was quite expected, is that the ants which we manually exposed to the disease had a much higher mortality, which was highly significant compared to their untreated nestmates. And overall, the survival of the untreated nestmate remained quite high. So we didn't have a massive epidemic where everybody died. Most of them remained alive. But despite this, we were able to detect an inflection point in the survival of the untreated workers around 4 days after treatment, where we found a hazard ratio, so a likelihood of dying, if you wish, which was 3 times higher than before these 4 days after treatment for the untreated workers. And 4 days sort of correspond to the time that it takes for the pathogen to be transferred from treating to untreated workers and to start killing the untreated workers. So we had some evidence that there had been some transmission of the pathogen in the colony, which had caused the death of some untreated individuals. But what we were really interested in finding out was whether the queen and nurses were less likely to die than the foragers, as was predicted by the organizational immunity hypothesis. So we compared the survival of foragers and nurses. Oh, I lost my screen. What's happened? Sorry. Can anyone hear me? Because everything went black on my side.
We can.
Yeah,
okay.
Everything.
I can't see my screen anymore. And I don't know what to do. That has never happened to me before.
Yeah, everything's active on our side. We see your video is as is, and your last slide.
Okay, I've lost control of my laptop, which has gone completely black, and I can only hear you. I might need to force restart. I'm really sorry.
No worries.
I just don't know how else to deal with this problem. This has never happened before.
If you need to pop off and then come back, that's fine. Let me pause the recording.
Okay. Okay, apologies for the issue. So what I was saying is that we compared the survival of the foragers and nurses among the untreated workers, and we found, as we expected, that the foragers had a higher mortality than the nurses, and the difference was significant. And as for the queen, none of them died during the 9 days of the experiment. So our first conclusion was that in agreement with this organizational immunity hypothesis, the queens and nurses survived better than foragers after a disease challenge where the disease were introduced on the foragers. But this could be due to a lot of factors. Maybe they have differences in their ability to survive the disease because their immune system is different. So we were really interested in knowing whether this difference was at least in part due to difference in exposure risk based on their location on the network of social interactions. So to try and disentangle these different aspects, we repeated the experiment with new colonies, of course, but this time we stopped tracking 1 day after exposure. And why did we choose 1 day? Because if you remember, I told you that during the first 24 hours after the initial exposure, the fungal spores are loosely attached to the cuticle of the individual. So that's a time where they can be transferred by direct contact with other individuals. And this is also a time where the fungus is still outside of the ant. It has not started multiplying, so it has not started an infection yet. And we therefore assume that the involvement of immune response mechanisms will be still at a minimum, so that what we can measure after these 24 hours will mostly reflect the exposure dynamic based on contact rather than other factors such as difference in immune function. So like I said, we stopped the tracking this time after 1 day, and then immediately we determined individual pathogen load using qPCR. So each individual was sampled, and we counted the number of spores that they had on their body with the idea that this should reflect how much they had been exposed to during these first 24 hours. And so we wanted to see whether contamination risk depended on network position. And first, of course, whether we found a difference between queen, nurses, and foragers in contamination risk, which we would expect if the structure of the network protects the queen and nurses from an infection coming from the forager. And this is where I explained to you what these black dots represent in these networks. These are actually the individuals which we exposed manually in our experiment. And so indeed, what we found was that the mean measured pathogen load on the queen and nurses was significantly lower than the one on the forager after 24 hours. So they had been exposed to less spores than the foragers. Second thing we wanted to do was to try and sort of link the individual load on each ant, which you can see as a color code on each of the nodes, as a function of their network distance to the manually exposed individuals, shown by a black dot. So the network distance is just a network measure that sort of measure how well connected two dots are. So two nodes that are connected via a direct link that is very thick, so that have many contacts, would have a low network distance, they're in close contact with one another, whereas two individuals that are connected via many intermediaries and very thin edges would have a high network distance because they're not directly connected. It goes via quite a long chain of intermediaries. So we wanted to check whether network distance was correlated with the pathogen load that we could measure on our ants. And that's indeed what we found. We found that there was a negative correlation between the pathogen load at 24 hours and the mean network distance to the manually treated individual, which sort of suggested that indeed the position on the network determines how much pathogen you're exposed to, so your contamination risk. And so presumably the better survival of queen and nurses were at least in part due to their lower contamination risk, which was due to their position on the network, isolated from these foragers that were bringing the disease in. The second bit we wanted to test about organizational immunity was this more global effect that this division into groups should slow down overall disease spread through this bottleneck effect. So the question was, does the overall topology of the network influence contamination risk through the colony? And so to do this, we try and determine whether there were associations between network properties and the prevalence and load of the pathogen at the end of these 24 hours, which we measured via qPCR. And we performed a partial least square regression to try and find the association between a variety of network properties, shows at the bottom, and measure prevalence or mean measure load on the untreated workers. And what we found is that 3 network properties were consistently associated with negative coefficient, suggested that they should slow down pathogen spread, because the higher this property, the lower the prevalence and the lower the load among untreated workers. And these properties were first modularity. So modularity of a network is just a measure that sort of measures the strength of division of the network into separate groups of community. Then the second measure was clustering, which is a measure of the node's tendency to form local clusters in which all the neighbors of one particular node are also connected to one another. Then the third property was diameter, which is the maximum distance between all pair of nodes. So I told you about network distance earlier. So here the diameter would be the maximum distance between all possible pairs of nodes. And these are properties that have been highlighted before by theoretical studies as transmission inhibiting property. So our experimental data sort of supported this theoretical study that basically a network which is compartmented into well-separated groups and that has a high heterogeneity should inhibit disease transmission. Now, on the other hand, we also found some properties which were consistently associated with a positive coefficient in the square regression, so that this property should favor transmission because the higher they are, the higher the prevalence and measure load in our colonies. Those were density, mean degree, and network efficiency. So in a network, the density is simply the proportion of existing connection among all possible connections. The mean degree is the mean number of connection of each node. And then network efficiency is just a global measure of the transmissivity of the network. So what we found basically is that a high network connectivity favors disease transmission, and this is also compatible with previous theoretical studies. So this analysis sort of gave the indication that indeed the global network properties seem to influence colony level contamination risk. But what we wanted to know was whether the structure of a reland network protected them against disease compared to what? What can we compare an observed network to? So to sort of figure out whether the structure of the observed network protects the ants, we actually compared these observed networks to randomized version of the network in which we conserved certain properties such as the degree of each node and the temporal sequence of interaction, but where we randomized the topology. So we randomized the partners. So basically we disrupted these preferential interactions within castes. And then we compared the properties of these randomized networks with those of the observed network. And what we found was that the observed network had significantly higher modularity and diameters than the random networks. So the reland networks have increased transmission inhibiting properties, but they also had a significantly lower density and mean degrees than the random network. So they have significantly reduced transmission favoring property. And this, just based on the analysis I presented before, suggests that these networks, these reland networks, should inhibit disease transmission compared to the randomized network. But that's just a prediction, and we wanted to test this further. So how can we test this? Well, obviously we can't measure transmission on a random network, so we are sort of obliged to use simulations. So we developed a simulation model in which for each interaction between two ants, we had a stochastic event of transfer of the pathogen from the infectious ant, so the one that carries a pathogen, to a receiver ant that does not carry the pathogen. And basically, the probability of transfer depended on the load of the infectious ant, the amount that was transferred depending on the difference in load between the two individuals, and the duration of the contact. And then we parameterized this model using experimental transmission data, which involved pairs of individuals for a very short time. So once we had our model, we basically validated it to check whether it meant anything. So we performed our simulation for our colonies that we had stopped tracking after 24 hours and for which we had quantified individual load. And we checked whether our simulated load were related to the actual measured load. And we indeed found that there was a good predictive value of our model with a good correlation between our simulations and the observation. The other way that we validated our model is that we compare the survival in our long-term experiment between workers that we predicted to receive a high load in our simulation and those which we had predicted to receive a low load in our simulation. And we found that our simulations were indeed successful at predicting survival, with the individuals that were predicted to have a high load dying significantly more than those which we predicted to have received a low load. So we can now use these simulation results to draw conclusions about the transmissivity of our network. And we perform a series of simulations in which the feed, so the source of transmission, were frequent foragers, so individuals that leave the nest often. And then we measured the simulated load on each individual and then calculated the mean. And what we found was that our observed network were associated with a significantly lower simulated load than the random, a decrease of around 15%. And this was not evenly spread across individuals, but it depended on whether you're a queen, a nurse, or a forager. So compared to the random network, the observed network had a significantly lower load for the queen and nurses by 42 and 28% respectively. And on the other hand, in the real-land network, foragers have actually a higher predicted simulated load than on the random network because of their close contact with the foragers. So we have this sort of selective protection of the queen and nurses, which are the more valuable ants in the colonies. The queen because she produces all the other ants, and the nurses because they're younger, and the foragers don't benefit from this protection. So overall, all these sort of little bits of information provide some indication for a form of prophylactic organizational immunity, whereby the structure of the network in normal conditions slows down disease transmission with a lower average load and confers proportionate protection to the queen and nurses from outside pathogens. We then looked into this idea of induced organizational immunity and whether we could find any changes in the network that would be induced by the detection of a pathogen by the ants, which could reinforce disease protection. And so for this, we repeated the experiment again with new colonies, again with this timeline of stopping the tracking after 1 day to focus on the time where the pathogen can be transferred and the ants aren't sick yet. And the idea was to compare the baseline network, so the network in healthy condition, with the network after exposure to the pathogen. So comparing the pre-treatment with the post-treatment network and trying to find differences between the two. And then we also wanted to compare control colonies, which were only exposed to the solvent, with pathogen-exposed colonies, which were exposed to the spores. So we could distinguish between changes due to our manipulation from changes due to the pathogen itself. And so all the graphs that I'm going to show you from now on will show the same sort of information, which is the changes in network induced by the pathogen relative to the changes in network induced by the control treatment. So positive value means a larger increase in the pathogen-induced colonies and in the control colonies, and negative value show a larger decrease in pathogen-exposed colonies and in sham-induced colonies. So what we found was that pathogen exposure induced an increase in modularity and in clustering. So there was a reinforcement of the transmission inhibiting properties of the network following exposure. And at the same time, it reduced the efficiency of the network. So we have a reduction in a transmission favoring property. The other property we had a look at is what we call task associativity. So what is task associativity? Well, if you look at a network in which you have different categories of individuals, such as foragers, nurses, and queens, associativity measures the degree to which nodes of the same type prefer to interact with one another rather than with nodes of a different type. So high associativity means that nurses really prefer to interact with nurses and foragers with foragers. And we found that pathogen exposure also increased task associativity of the network. So basically increases segregation between nurses and foragers. So based on this topology analysis, we would just assume that the changes induced by the pathogen that we detected in the network should reduce disease transmission. But again, we wanted to test that a bit more formally, and we used our simulations. And because it's not possible, why did we do that? Because simply it's not possible to measure transmission of a pathogen on a healthy network, because as soon as you introduce the pathogen, the ants react and change the network. So we're sort of obliged to do this via simulation. So we performed this simulation on the pre-treatment network and on the post-treatment network, and we tried to see whether we found differences in the amount of spores that spread among the untreated workers. And this first shows you the mean simulated load among untreated workers, and again, the change induced by the pathogen exposure compared to the sham exposure. And so we found that overall, the changes in network induced by pathogen exposure led to a decrease in around 30% of the simulated load among the untreated workers. So we do predict that it should reduce transmission among individuals. And then looking at different casts, we calculated the probability of receiving a high versus a low load. And just to remind you, the high load was a load that we know lead to a higher mortality than the low load based on our sort of combination of simulation and long-term survival data. And what we found was that the queen and nurses had a significant decrease in their probability of receiving a high, potentially mortal dose, which was induced by the change in the network. So a decrease in mortality risk for the queen and nurses, but not for the foragers. But at the same time, they had an increase in their probability of receiving some pathogen, but at a very low dose. And this, based on other studies, is expected to be positive as well, because it should induce a form of immunization, so protection against later exposure to the same pathogen, a little bit like what vaccination does in humans. So we have a double benefit for the queen and nurses of the changes in the network induced by the pathogen. So again, these sort of information indicate that yes, there are changes in the network induced by the pathogen, and they tend to reinforce protection against disease. So it does seem that there's an induced form of organizational immunity. So I can see I'm reaching the end of my time. Do I have 5 minutes more since I had that interruption, or is that okay? Yeah. Okay, then I'll continue. Our last bit was to try and detect which changes in individual behavior were responsible for the changes in network which we detected after exposure to a pathogen. The first thing we had to look at was the proportion of time that individuals spent outside. You can see here the nurses on the left, the untreated foragers in the middle, and the treated foragers on the right. And we found that both the treated and the untreated foragers increased the amount of time they spent outside, and consequently, they tended to increase their distance to the center of gravity of the colony. Now, it's tempting to sort of infer that the treated foragers isolated themselves from the colony by spending more time outside. But at the same time, the untreated ants also did so. So is this a form of proactive distancing between foragers and nurses, or is this due to caregiving, where the untreated foragers try to protect the treated foragers, and as they're outside, they then spend more time outside? That's not very clear, but it's sort of interesting to think about. We also found a change in the relationship between workers and brood, with nurses increasing their overlap with the brood, while treated foragers decrease their overlap with the brood. And overall, the brood location changed, with the ants moving the brood further inside the colony, although this was by a very small amount. So again, it's tempted to think that perhaps the nurses spent more time on the brood because they were trying to protect the brood and removing the spore from the brood, so doing some caregiving. And while again, the treated foragers just isolate themselves from the valuable individual in the nest, and perhaps moving the brood is a way to protect, to avoid sort of contact with the infectious sources. Regardless of this interpretation, what we found was a spatial reorganization of the colony. And that spatial reorganization of the colony led to changes in social interaction. So in particular, we found that both nurses and untreated foragers decreased their contact with the treated foragers. But we also found changes in the interactions between the untreated ends, so the ones that had not been exposed experimentally, with a decrease in contacts between nurses and untreated foragers. So overall, the changes in social contact induced by the changes in behavior. And again, it's tempted to either sort of interpret these things with, on the left, the fact that nurses and untreated foragers decrease their contact with infectious individuals, could be a form of isolation by the treated foragers or avoidance of these foragers. And then the increase in distance between nurses and untreated foragers could be a form of proactive social distancing. So overall, we found that the network changes induced by the pathogen were due to changes in the behavior, not only by the treated pathogen-exposed workers, but also by the untreated workers, which were able to detect the presence of the pathogen and react. And this is just a way to sort of summarize what we found. So we have the pre-exposure network of the left and the post-exposure network on the right, with the exposed individuals this time in gray, and can see perhaps three processes taking place. One being self-isolation, with the pathogen-exposed individual removing themselves away from the colony and increasing their distance from the rest. Then you also see that there's some caretakers, so a few individuals, typically foragers, that remain in close contact with the pathogen-exposed workers, potentially because they're caring for them, trying to remove the spores from their body. And at the same time, you see a form of proactive distancing between the bulk of the colony and those caretakers, so an increase in the distance between the untreated foragers and the untreated nurses. So to conclude, I hope I've convinced you that societies and societies have non-random interaction networks. And regardless of the cause for this, of the evolutionary pressures that led to these non-random interaction networks, one of the consequences of this is that this confers a protection against pathogen transmission, both because the division into sort of separate groups causes bottlenecks effect that slow down overall disease transmission, and because the queen and nurses, which are more valuable for the ants, are isolated from the foragers, who are more likely to pick up an infection from the outside. And we also found that ants use network plasticity as a fairly low cost, effective and early response to infectious pathogens. So they reorganize their network, and overall, this further reinforces the protection against epidemics that the network confers them. So question could be whether this might be more widespread than we think. And with this, I'd like to thank you very much for your attention. I'll be happy to answer questions.
Okay, thank you, Natalie. I'm going to be handling questions. So the procedure for that is just you can either raise your hand as an attendee or type in your question into the Q&A. Okay, and I will unmute who has ever raised their hand. So Peter, yes. Go ahead, you're unmuted. Oh wait, you can unmute yourself.
That's the way I like it, to be able to unmute myself.
Yeah.
Right? So yes. So I enjoyed it, and I have to say it gives a lot of evidence for the fact that the ant colonies are keeping the transmission from spreading within them. But I'm thinking it from the standpoint of the pathogen in the fungus. Wouldn't it be also advantageous for the fungus to have the infection be chronic rather than acute? So in other words, if it actually spread very rapidly through the colony and killed the queen, that wouldn't be a long-term advantage to the fungus, as opposed to having the queen and a lot of workers survive and continually be, providing bodies to be infected, and therefore be able to transmit not so much within the colony, but across colonies. Again, if you have a lot of chronically infected colonies, that might help the fungus spread from colony to colony and do well in the environment. So that's my question.
Okay, so I would agree with you if this was not an obligate killer pathogen. But for this pathogen to transfer to other colonies, it has to kill the host. It's only when it kills the host that there will be sporulation and production of new conidia that can then transfer to other hosts. So there can't be a sort of continual low-level infection with a continued production of spores. What was studied here was this sort of initial process when an ant comes back with some spores, and there can be a dilution process, a dilution sort of effect through the colony before the fungus enters the ant. But once the fungus has entered the ant, after this 24 hours, either it will kill the ant and be able to transfer, or that's a dead end for it. So there's no sort of option for this particular pathogen to stay at low level and benefit from it to transfer to others.
Wouldn't it still, I mean, even though it's killing some proportion of the colony, as long as it's not killing the colony all very rapidly, then it's still sort of producing the spores that could be dispersed in the environment. Again, if you just hypothetically said it, well, if it within 24 hours, if it killed the queen, then its lifespan within that colony is going to be very short as well, as opposed to just sort of hanging on the edges and killing workers some proportion, sporulating over time. It would have higher reproductive success.
The way I understand it, though, is when you're exposed, either you sort of get on top of the infection, and then that's the end for the fungus, or you die and you can transfer. There's no midway of long-term infection that can stay with this colony, because there's no way for this to sort of be maintained, since there's no transmission if there's no killing. Does that make sense?
Yeah, I guess I'm just arguing that you don't want to kill everybody. Especially you don't want to kill the queen, who's the goose producing the golden eggs for future workers to be infected.
But either there's an infection that occurs immediately, or trying to think. Typically, these workers as well, when they die, before they die, they will sort of realize that they're sick, and then they self-isolate. That in a way that we couldn't test in our experiment. They actually go very far away from the colony and die where they can't infect anyone else from their own colony.
But that would be a very wonderful strategy for the fungus too. So
yes.
Right.
Yes, we know it's ant motivated because they do the same when they've been poisoned with a toxic or CO2. They sort of, when they reach the end of their life, they just go far away. So we believe it's the ant's decision rather than a manipulation by the host. And the other thing I would say in this particular system is that because it's a generous fungus that also infects a variety of solitary insects, I don't think it would necessarily be sort of adapted to infecting a social insect colony and have particular strategies for that.
Great. Okay, I'm going to move on so we have some other questions that can get responded to. The next question is from Noah Pinter-Woolman. Can you, may have missed this, but I don't think you mentioned it, how colony size might affect the colony response to the pathogen?
Right, that's what we're studying at the moment. We did not set out to study that in that particular study, but we believe that so there's both different risk of transmission depending on colony size, and also cost benefit ratio depending on colony size. So presumably a colony that's got 10 individuals, the survival of each particular individual will be very important. So perhaps they will favor caregiving and really try to save individuals, because if they lose one, this could be very major. But at the same time, they might prefer to exclude that individual to avoid the epidemics, because they don't yet benefit from this organizational immunity, which is supposed to get stronger as the colony grows and division of labor becomes stronger. So basically, we're running an experiment at the moment to try and see the parties effect and see how colony size may affect these strategies.
Great. If we have time, I have a question. First of all, great, really interesting work. I was curious though, if you know anything about what individual ants are aware of. And I probably can't answer this question, but I think it's an interesting thought experiment. Like, do these infected individuals, I mean, presumably they're aware of their infection state, but what is the, how aware are the other ants that ant is interacting with? And so I can see, you know, the ones that these caregivers, you say, probably have some, you know, have detected the pathogen, but for the rest of the colony to say self-isolate, I wonder if there's some kind of signal that's being sent to everybody else, or if that is like an epiphenomenon of something else, some other process happening, and if that's something that you are investigating.
Yes, so I think a lot of what we found could be explained as an epiphenomenon. So we know that by direct contact, they can tell whether an individual is infected or not. We know that because when you have individual in pairs and you expose one individual, the others immediately increase their grooming. So we know they can tell there's spores on the cuticle of the other one. We also see that the exposed individuals immediately increase self-grooming. So they know they've got spores on their bodies. And this can be maybe, they can detect that maybe due to the smell, or maybe there's a tactile cue from, you know, being all sorts of grainy from carrying the pathogen. So we know they know when they come in contact. Now, about the sort of longer distance effect, I'm saying perhaps it's an epiphenomenon, and I tried to quickly touch upon that during the talk, but I was trying to rush to sort of stick to schedule. If it's the case that the treated individuals self-isolate because they know they're a danger to the other, and then the untreated foragers find them, realize they're sick, and start caring for them, then they would automatically spend more time outside. And then the network would show this as an increased distance between nurses and foragers without nurses needing to sort of change their own behavior. And then, so that could be an epiphenomenon. At the same time, we see the nurses spend more time on the brood and the brood being moved. So we think they know something's up. And I think the reason there may again be quite simple, because some of the treated foragers will still enter the nest. Just they increase their time outside, but they still go in. And while doing so, they're still transferring some spores, perhaps on the brood, and that's what the nurses are reacting to. They see that some of the brood have spores on them, and they react to that. This being said, recently I've been looking at what the ants do just after being infected, and I've noticed them producing an alarm vibratory signal, not unlike termites. So, and then they all shake. And so I do wonder whether that might be something that's happening that also is used to transfer the information longer distance. But I would not want to make any strong claim on that, because this is just anecdotal for now. But so perhaps there is also a little bit of communication that it sort of informs the untreated workers far away.
Cool, thank you. There's one more question that Nitika has, if we can please squeeze that in.
Yeah, sorry, we might be running out of time, but I just wanted to ask this question. First of all, amazing talk, very exciting results. So the question I had was, how do you expect the social networks and thus the transmissions to vary across species, in the sense that some of them might be cavity dwelling and might just be confined to a small space, while others might have, chambers and tunnels that sort of spatially segregate them. So basically, how does architecture play into the constraints of social contact?
Again, I'm sure it's got a huge influence, and the experiment we ran wasn't designed to test with it, but we're looking into it at the moment, trying to see how architecture influences the overall transmission and whether they can use this as a reaction, and they sort of segregate between the chamber a bit more. this is a question that we want to go into in the future, basically. But I'm convinced that nest architecture has a very big influence indeed.
Thanks.
Okay, I don't see any other questions. So I guess we'll just take this moment to thank Natalie for coming, and thank you for such a wonderful talk despite the technical difficulties. It was
sorry about that.
No problem. It was fantastic anyway. You handled it very smoothly. So I guess we'll look forward to meeting with you, and hope you enjoy your virtual visit.
Thank you very much.